Um, so, yeah, I've been since 2007. I've been most of my research has been about causality. So I think there are many problems in causality that I could talk about today, but I just selected one particular problem question. I would like to talk about going towards reliable and data-driven approaches to what you could call scientific or causal discovery. So just to get us on the same page, I'm not sure if that's necessary, but I would say that if we have two variables A and B, then A causes B if an external intervention on a system that changes A also results in a change of B. But with this sort of starting point, you can still wonder how to model causality mathematically, how to model causality and uncertainty, so to do statistical causal modeling, how to discover causal relations from data, how to estimate their strength, how to use this knowledge for making predictions, and how to exploit the knowledge when you want to control or optimize something. So that's very broadly the research that's done also in my team. I've met several PhD students and postdocs. One very interesting class of methods that have been proposed are so-called constraint-based causal discovery methods. And in 2000, it was shown that even without doing experiments, so from using purely observational data, you might be able to draw causal conclusions. And the basic idea here is that we start with some data, we run conditional independence tests to look for certain statistical patterns in the data, and then we reason back about the possible causal graphs that are compatible with these patterns. And nowadays, many algorithms for this purpose exist, and they can deal, they are very generally applicable. So they can deal with latent confounders, with selection bias, with causal cycles, and they combine various data sources. So here's one practical problem that I've been working on for quite a while already. I think I started around 2011 on this particular data set, but it still keeps me busy. This is a data set where biologists measured protein expression levels in individual cells of the human immune system. So here you see 8 different experimental conditions where they added different reagents to these cells. So they perturbed them in certain ways. And on the here you see on the x-axis 11 different proteins, and in color you see their activity. So you can see that the activity changes depending on which reagent was added, and also that there is some variation between the cells. Now, one task that is of interest here is, can we understand what biologists call the signaling network and what we would call the causal graph of this system? So how these different expressions of these proteins interact. This has been kind of a problem since 2005 in the field, and many methods have been used to analyze this data. And here is one of the results that I got myself recently. And well, it looks nice. So you see some kind of causal relations that are discovered, but at the end, you don't really know what the true causal structure is. So you can come up with many graphs that look like this, but then at the end, you always have this problem, like, can we validate this? So I would like to move on to another problem, which is also in biology. It's about the discovery of gene regulatory networks. So the basic idea is somewhat similar. We have data, some observational data of yeast gene expression levels. Now there are about more than 6,000 of these genes, and they all come with their own expression. These have been measured in 260 observational samples, but also in 1,500 interventional samples, where in each case, in each sample, one particular gene was knocked out. So it was disabled. And then you can see the effect on the expressions of the other genes. So the challenge here is whether we can, in a purely data-driven way, so without using biological knowledge, predict which genes would strongly change their expression when we knock out a given gene. So to find the strong causal relations between these gene expressions. And it becomes particularly challenging if we are not allowed to use any data corresponding to that particular knockout experiment to make the prediction. And that's the nice thing here of this data set is that we can, if we don't use this data for prediction, we can still use it for validation. So we can use the same data set for both purposes. So here's just an example of how this would look like. Here you see two genes on the X and Y axis, and here you see the measurements. The blue dots are the observational ones, and the red ones are the interventional ones. And this is on the left an example of a causal relation, where if we knock out the gene that's on the x-axis, so its expression goes down a lot, then as a result also the expression of the gene on the y-axis is reduced by quite a bit. So that's a causation. On the right-hand side, you see a non-causal relation, where if you knock out the gene on the x-axis, it has no effect on the gene on the y-axis. So the question is, can we distinguish these? Here, causal discovery algorithms can indeed do this quite remarkably. So you can see, I'm not going in the interest of time to explain you exactly what you see in these plots here, but basically it shows you that some of these algorithms, also invariant causal prediction that was already mentioned before, but other simpler algorithms can actually find or distinguish these causations from the correlations in this data, at least for the first 100 or so pairs of genes, and for the other millions it doesn't work that well anymore, unfortunately. But it's at least a large-scale validation of this methodology. So basically the general problem statement I would like to pose here is, even though causal discovery methods work provably in theory and also empirically in simulations, how can we improve their reliability in practice in order to enable this promised data-driven approach to scientific discovery in biology, but maybe also in other disciplines? Thank you.
Okay, thank you very much, Joris. Another very interesting presentation and going in yet another direction.